Okay, so what we're going to do next is we're going to talk about some alternatives when it comes to different types of designs. So far we've been talking about independent group designs. Now we're going to talk about matched and repeated measures designs. So once again, we think of independent or between subjects designs as conditions where different people are in different conditions. So you have an experimental group and a control group. And once again, you can have different variations where, for example, you might have a pre-test and a post-test, or you might have more than just two groups, but there are still independent groups of people going through different things. We're also going to learn about matched pairs designs and within subjects designs as alternative experimental designs using slightly different methods for different reasons. So matched pairs designs are when you have very small samples of people and random assignment wouldn't be as effective. As a reminder, when we were talking about the deck of cards activity, random assignment works best when you have a large sample size. And when your sample size is too small and you want to really make sure that your groups differ on, that your groups start off as similar to one another rather on important qualities and that those similarities are spread out between the two groups, sometimes you have to be in control of that yourself instead of making randomization do the work for you. So for example, if you wanted to make sure that the two groups were matched on things like gender or additional qualities like that you believed would be important, like for example, whether someone had a learning disability for reading, then what you would want to do is match people on important characteristics and basically come up with a pair of people that are similar to one another. So for example, I might want to have male A with a learning disability and male B with the same learning disability as a pair. And I might have female A matched with female B, and those females might not have learning disabilities. So you would pretest on important demographic characteristics like gender and learning disability if, once again, that was something that was very important to make sure to spread out evenly among your groups, and then you'd form matched pairs. And then you'd randomly assign one of the people in that pair to the experimental condition and the other one to the control condition. So this assumes that the groups are equivalent to one another at the start on things that you think are important to control for, but they may not be equivalent on factors that you didn't measure. So this is an imperfect solution when you can't have effective random assignment and you really want to make sure that the two groups are roughly similar on important characteristics that you're trying to control for. Repeated measures is when the same people see all the different conditions. So for an example, I did an fMRI study, which is actually very common to use repeated measures for, which I'll get to in a little bit. And in that study, I exposed people to high and low status conditions. Now, in this particular study, participants were exposed to multiple people who were high status and multiple people who were low status. So I tested ahead of time in pilot testing to determine things like attractiveness and whether certain phrases would be associated more with high versus low status cues. And so people would see 12 people who were high status and also 12 people who were low status intermixed with one another in the MRI machine. So everyone was seeing 24 stimuli, half of which were high status and half of which were low status. On a side note, I actually did make it so that it was high and low status based on gender. So males saw high and low status males and females saw high and low status females. Another example would be in a jealousy study that I did. I brought people into the lab and made them think that their romantic partner had flirted a little bit with a potential romantic rival. So they went through a chat activity, and then I showed them the chat transcript, and in the chat that transcript, it looked like there was flirting going on. I actually copied and pasted in slightly flirtatious dialogue as part of that. And then I said, okay, here's the chat transcript. Now you get to play a game in the next part of this study. And this game was an aggressive game where you got to blast the other person with sound if they lost the round, which is considered to be a fairly standard measure of aggression. And I said, okay, you can play this game, and you're going to be playing this game against your partner first. And then you're going to be playing the game against your rival. Or I told people you're going to be playing against your rival first, and then you're going to be playing the game against your partner. The reason why I wanted to do it that way was because I wanted to see who people were aggressive towards, if they were selective about their aggressive behavior, or if they just lashed out at the first person they were exposed to. But in this particular case, all people played both games. They played the game against the partner, and they also played a game against the rival. Now, the fact that I mixed up the order is a specific solution to a problem that we'll get to in just a little bit. And once again, my particular concern was I wanted to explore whether people would be aggressive against the first person they played the game against, or if they would actually pick and choose who they would be aggressive against, whether the person's role, partner or rival, would actually matter. And it turned out that it did. Spoiler alert. So when we look at some advantages and disadvantages of within group designs, one of the advantages is that it's very efficient. You don't need as many people because all participants are in all conditions. So if, for example, you had 4 conditions, in a classic study, you might need 25 people in each of your conditions. In this particular case, all of your participants are experiencing all 4. So it's much more efficient model. It's also incredibly cost effective when you have very expensive equipment like MRI machines, or the participants require extensive training, or you're doing something that's of higher risk that requires screening like drug testing. It's also very often used in animal research where you have to be very efficient with how many animals you're making use of as part of the ethics requirements for your study. One of the problems is it can be more time consuming for individual participants, especially if it's a study where they have to come back after for a second round or for a second day. Another distinct advantage to this type of study is that you have greater statistical power. Even considering that you are running less people, you actually have a greater ability to detect statistical significance with a within participant design because there's less variability across conditions due to individual differences. You don't have a separate pot of people in condition A and condition B. It's the same people in both conditions, and as a result, individual differences or error, which is the denominator of your test, is going to be a lot smaller. You have less error, less individual difference, and as a result, less variability, which means you have a bigger result. So there's more power with a within participant design. There should be some review from statistics. Hopefully you remember our conversations about power. But regardless, within participant designs are both more efficient and they have higher power. One of the challenges is that it can be difficult to generalize your results. What I mean by that is that in the real world, we don't tend to encounter multiple conditions. Usually you encounter one or the other. This exposes you to all of them, which is just a little bit less realistic. Another challenge is that you can't really use this design when people are exposed to something that might lead to a permanent change. So if you're studying a surgical intervention, for example, you can't say, well, okay, so I'm going to do the surgical intervention and then we're going to take a break and then we're going to come back and not do the surgical intervention. It doesn't really work that way. So while within participant designs can be really efficient and effective, they are not necessarily always possible given sometimes your experiment and the way that your conditions work. One of the other distinct challenges that we can encounter when it comes to Within participant designs, keep in mind every participant experiences every condition. So for example, if we had intervention A, intervention B, a control, or drug dosage A, drug dosage B, control, therapy A, therapy B, you kind of get the idea. Stimulus A, stimulus B, every participant would be experiencing both of those things, or sometimes if there's multiple stimuli, all 4 or all 10 of those things. So when you have multiple exposures to multiple stimuli, sometimes one of the challenges is order effects. There's different types of order effects. Sometimes people can improve due to the practice that they gain on the first time that they do the task, so that they just get better and better over time because of that initial exposure. Sometimes multiple exposures to stimuli can lead to fatigue, where there's deterioration in performance or in reaction due to being tired, bored, or desensitized. That was a problem we encountered in fMRI research. We had to mix up the order a little bit so it wouldn't be anticipated. So for example, I couldn't go high status, low status, high status, low status, high status, low status, because then they'd know what the next picture was going to be. I had to kind of mix it up a little bit. And there was actually a formula I used for how to make sure that the order would not lead to desensitization. And then there's carryover effects where the treatment of the first treatment lingers and continues to impact subsequent treatment. So for example, if you're taking a drug, you could still be under the effects of that drug when you participate in the second session. And so it's important to make sure that the effects of that particular drug are out of your system before you move on to the next level of dosage or the next type of drug intervention. There are 2 different solutions to order effects. The first solution is counterbalancing, and counterbalancing is similar to what I did with the jealousy study, where I had half of the people play the aggressive game against their partner first, and I had the other half play the game against the rival first. With counterbalancing, you randomly assign people to different orders of the stimuli. And keep in mind, every person experiences all stimuli. So let's go ahead and go through an exercise where there are 3 stimuli, A, B, and C. Or, you can think about this in terms of different levels of drug dosage or different levels of intervention or something like that. But let's go ahead and think about it as an fMRI study where there are 3 different types of stimuli people can be exposed to, A, B, and C. In this particular case, you would want all possible orders randomly assigned. So some people would see the order ABC, some would see ACB, some would say BAC, and so on. So you can see here's every single possible combination of stimuli in every order, and you would randomly assign people to one of these 6 different orders so that each person would have a different order that they were exposed to, so that you could actually test what was going on and compare the different orders to see if the order matters. Ideally, of course, you'd want there to be no order effects, and if there was no order effects, there would be no difference across all 6 conditions. And then you could further test if there was a difference between A versus B versus C. So you'd want to just make sure that there's no potential order effects, and you can test that this way. This can sometimes be challenging if you only have a limited number of participants and multiple conditions. So for example, when you have 3 stimuli or conditions, there are 6 possible combinations. If you have 4 stimuli or conditions, there are 24 combinations. If you have 5 conditions, you have 120. And if it's important that you have more than 1 participant in each possible combination, then all of a sudden you start ballooning the number of participants you need to run. So one solution to that is what we call Latin squares. Latin squares presents a limited set of possible combinations where each condition appears in each possible ordinal location once. I'll go through an example where I illustrate this. And each condition follows and precedes each condition once. So here's an example of 4. And if you'll remember, going back to the earlier slide, when you have 4 stimuli, the number of possible permutations goes up to 24. But here in the Latin squares, you only need 4 combinations to meet these requirements. So for example, let's go ahead and take a look at B. B occurs second here, first here, third here, and fourth here. So it's in every possible ordinal location. B is after A here and before C here. It's before D here. It's after C and before A here. And it's after D here. So you can see how it's before or after A, C, and D in these different rows. And this occurs for all of these different letters. So each of them occur first, second, third, or fourth, and each of them occur before one of the other conditions and after it at least once. So this is an efficient way to have the different order effects explored using a Latin squares, especially when you have multiple stimuli and a limited number of participants. Our second solution is a time interval between treatments. So one of the things that you can do, remember I was talking about using different drug dosage levels or different types of drugs and making sure the drug was out of your system. You would just have enough time in between to make sure that the effects of your first stimuli had worn off. There's different ways that we do this. So for example, with fMRI studies, you might have a little cross in the middle of the screen that people focus on, which is known as a fixation point, and that can help kind of reset things a little bit between stimuli. Sometimes if people are doing different tasks and you want to make sure that one task doesn't bleed over into the second, then you could have a distraction task in between. So for example, you could say, count down from 1,358 by intervals of 7, which makes people think really hard and distracts them enough so that they kind of forget a little bit about what they just went through earlier and prevents that carryover effect from happening. Or once again, you literally let enough time pass for the drug or condition to wear off. So if you're looking at animal behavior, for example, you might want to wait until extinction occurs. Or if you're doing a drug study, you wait until all the drug is out of someone's system before you study the next condition.